Thank you, and I'd like to thank the organizers for inviting us. This is joint work with Dr. Barry Duesman of the New York State Police. We've been collaborating for almost 10 years on this project. And to use another meaning of the phrase, there's currently a crushing weight of DNA evidence. The amount of DNA quantity just keeps going up and up with property crime, touch DNA, and demands on laboratories. And what some people perceive as the DNA quality, with mixed samples, degraded samples, low level, and so on, the quality seems to be going down, and this leads to some DNA problems, backlogs, low information yield, and the crushing weight leads to possibly a long wait for evidence. And certainly in the US, we're seeing more and more criteria for when we just don't analyze the data at all and move on to something else. And that's a very long wait indeed. So TrueAllele Casework provides computer interpretation of DNA evidence, and as some of you may have heard yesterday, it mathematically models the STR process. The primary goals of the project with New York and many other groups is to achieve objectivity in DNA interpretation, ease of use in being able to get a interpretation happening for you by machine. Information is a significant goal. We want as much information yield as possible from the data that is being brought into the lab. We don't want to throw anything out if it contains information. And the focus of this talk is productivity, and these are the same 4 terms that Barry uses in his talks all the time about TrueAllele. In this case, with productivity, we'll measure how much work might be done redundantly that could be avoided. So let me go through each of these 4 points. Let's begin with objectivity. Objectivity is inherent in the interpretation of evidence by a computer using Bayesian inference of genotypes, because you begin with the DNA evidence, an interpretation occurs, and you end up with the DNA evidence genotype. You've never seen the suspect, you've only seen crime scene DNA evidence. It might be 1 sample, it might be 10, it might involve elimination samples, victims, whatever it is, it doesn't involve suspects. And then a match is made to suspects. 1, 2, 10, 100,000, doesn't make any difference, you've already locked that genotype into the database of what your answer was. And when that match is made, as you see on the right, when you combine an evidence genotype together with a suspect genotype and a population genotype, you have a DNA max score or likelihood ratio, and the result is an objective computer interpretation of the quantitative data. Let me move to the next point, which is ease of use. In TrueAllele, what a user does is they ask questions. If it's a routine process, like volume crime, you can upload 200 questions simultaneously from one plate, ask it to solve everything for 1 and 2 unknowns from 1 amp, and the system happily goes off and processes. For casework, workers typically prefer, we certainly do when we get into the data, to instead maybe take 2 or 3 minutes and ask a question about the data in a case. And if it's an interesting case, like, it's 40 picograms of DNA, then we often ask several questions to see how much information we can get. So after the DNA comes off the sequencer, it's uploaded by a plate into a database, and then if you're going to go through this process, you come into an interface, and the request interface in the viewer program, and you say, please give me all the data that's associated with my case. You do it by name, it finds it. Then you're looking at the first panel on the left. There's your data. The top 2 coded by the user and the computer in the interface with a darker blue showing profiler plus and cofiler as evidence, and then underneath it there's, for this purpose, irrelevant suspect. It's not going to be involved in the request. The user then says, I'd like to analyze an item of evidence. You see in the middle panel on the top there's an icon for the DNA for the evidence, and then from that the user says, well, in this case, because I'm going to continue with this example, let's analyze it as 2 unknown. There are all sorts of questions you could ask, and this is what you see. You might also want to analyze a suspect, so the matching within the case after genotype inference happens automatically. A trained user would do this in about 2 minutes. And if it's an interesting case, you have to stop people from going back and asking more questions. It's a fun calculator. The computer goes off and processes. How does it do that? We run parallel systems. In our office, our machine has 24 processors. We're adding another 8 next week because we're doing more validation projects with people. And currently, over about an hour, we'll see, well, actually the easiest way for us to measure it is that we know that every day we crank out about 300 cases from the 24 processors, typically mixtures and low-level and interesting things. That's how we can schedule our work. And so we tend to solve it with parallelism because computers are cheap. When we're done, you have an interface with a whole bunch of things you could look at to review the data, and you get answers. We saw this yesterday. This is what the mixture distributions look like. We see on 0 to 100% on the x-axis, and here are two distributions, about 40% and 60%. The minor contributor is in blue, and we'll continue looking at that. A genotype is inferred. What you're seeing in every row is another locus, and to the right is its probability distribution. All mixture interpretation, anything that's uncertain, is always interpreted and produces a probability distribution. What else could it be? It wouldn't be science if it weren't. Certainly RMNE, CLR, everything produces a probability distribution over the allele pairs. So this is a genotype. When you get a lot of bars to the right and you're getting closer on that 0 to 100% probability scale to something that looks like a definite allele pairs trying to come out, it means you're going to get more information typically, and it was, for the computer, an easier sort of case to resolve. So on this minor contributor, I'm going to quickly describe this again. This is a picture where you're seeing the base pair on the x-axis, and you're seeing the RFU on the y-axis. And the data are in black for D13, and there's a cartoon drawn over it by the computer. All the other stuff, the lettering and so on, is just PowerPoint saying what the genotypes, the allele pairs in D13 are. What the computer is showing you is that, well, suppose you want to know what happens with roughly 60-40 of the orange contributor to the blue contributor with those allele pairs. You can choose them. The computer certainly is considering everything that could possibly exist in its calculation. Then it brings in stutter. I guess you can see the stutter all the way on the left, on the bottom. I can point to that, I guess. You can see the relative amplification, given where the peaks are. There's sort of a skewing. It creates a pattern that you see in gray, and it compares the quantitative pattern. It does tens or hundreds of thousands of these proposals against the data, and those patterns that quantitatively better fit the data tend to have a higher likelihood and give a higher probability. Notice that this might be very evident visually to people and to a quantitative computer, but if you had a threshold much over 100, you'd start throwing out what you were looking for. In the reporting, we have another interface that does a lot of reporting. It's to give you a snapshot. We use it for validations. We sometimes will be looking at 100 things at once here. On the top, you can see, you can set what theta value you want for co-ancestry. You can look at this on a log scale or linear scale. You can look at every locus individually or look at them jointly. And what we're seeing here is you can look at any number of evidence genotypes against any number of controls with any number of populations. To simplify, let's look at the one minor contributor genotype that we just saw, and for what the total likelihood ratio is, log likelihood ratio information. We'll have two different comparisons, a suspect and an elimination profile. Elimination doesn't match, it's moving over to the left. The scale over here is -20 to +20 log units. That's, you might call, an exclusion. And on the right, you're seeing something that's over in the 15 to 20 range, and this is showing it for two different ethnic groups. This is the interface where people have the most fun because they get to try out what does the answer mean, whether it's for a case or for a validation study. And in the report interface, we also can generate detailed calculations and reports that are useful for the case folder. This is a more detailed one. It gives a likelihood ratio statement for every locus. It will give a lot of information, including the genotype, which of course is a probability distribution over allele pairs. And it gives the likelihood ratio both in linear and log form. This is sometimes useful if you want to establish how a calculation was done. Okay, so I'd like to move on now to information and productivity, now that you have a sense of what the system looks like. So in this study, collaboratively with New York State, you'll see all the different people who participated at the end on the acknowledgement slide. We gathered together a lot of data. It took about a year to gather the data. It took about another year to get permission. It took another 6 months to get permission to submit the paper. This has been a very long study. But there are 368 evidence items that range the full spectrum of what the New York State lab sees, as you can read here. I want to quickly show some pictures of what results are. What we're seeing here are 8 cases. That's at each point on the x-axis is another case. And these were cases for which New York State looked at as a 2-person mixture, and they had a victim profile. So we'll see in a second what they did with it, but they did a CLR analysis, assuming the victim. The vertical scale is information, log likelihood ratio, going up from 0 to 25. So for each of these, we did these in duplicate, and blue is the first computer run, green is the second computer run, and we measured reproducibility. John Butler's shown that with U.S. labs, reproducibility on 2-person mixtures ranges over 10 orders of magnitude. When you have consider population variation, typically it's plus or minus 1 log unit in the U.S. as opposed to 10. And the variation that we see in TrueAllele in simple 2-person mixtures like this is in the order of hundredths or tenths of a log unit. So it's a very reproducible process. Now let's see how we lose information. So in blue again, on the same scale for the same 8 cases, the 1 unknown 2-person mixture, this is what TrueAllele did for each case, with an average of about 10 to the 17th likelihood ratio. Human review, using the usual sorts of thresholds and equal likelihood assignment to allele pairs instead of based on quantitative data, lost about 4.5 log units, and you see case by case the drop to where the orange bars are. When we go to 2 unknowns, this is RMNE is what people were doing. So again, these are all cases for which New York State reported a statistic. I'll change it in just a second. So these are 8 different cases now where they did not have a victim reference, and there's less information. You can see the computer drop from maybe 17 log units to 13, but the human review, relative to the gold standard of a quantitative interpretation, lost over 6 log units if you look at the average drop from the blue bars of the computer case by case to the human review. Typically 10 to the 6th, 10 to the 7th, which you see in the right in orange, is what a normal RMNE would be like in the States on 13 loci. And the information loss here is due to the fact that you're not using a victim genotype and you're using equal likelihood assignment to all possible allele pairs, which diffuses the probability. Okay, so now we've seen when New York State got a score, we lose more information as the cases get more interesting with less information. What happens when we look at all 85 or so mixtures in this case? This is probably the most interesting slide that we got from the whole study, and so I'm going to spend a minute or two on it. What you're seeing on the x-axis is just a listing of the 85 evidence items, and they're ranked by decreasing information, that blue edge, as determined by TrueAllele interpretation. So they're sorted. That was the most, 10 to the 20th, going down to maybe 10 to the 3rd. The median information value in the center is about 10 to the 15th, 10 quadrillion to 1 as a likelihood ratio. And so the height of that curve is telling you what the log likelihood ratio information is for each interpretive case, and the area of that blue is telling you what the total information extracted across all the cases was for the study. Now, this is what people did in these bars. The best results are the 4 gray RMNPs, where you apply thresholds and pretend the mixture single source. Fantastic results. You just get the full answer. It was supposed to be funny, but I guess it's not. Okay. In green, we then have 8 where the victim was known, a CLR was done, and you get a reasonable amount of information. And then there were 12 in orange that were RMNEs or CPIs, where you lose more information. So here are the 2 most interesting facts about this particular picture. The first is, if you look along the x-axis and ask how many of the cases for which, actually all the cases for which the computer got an answer, what fraction of the case of the mixture items that were done, was there any reported likelihood ratio result at all? And the answer is less than 30%, fewer than 30%. So we're going to talk about that in a second, but that implies that there's a whole host of information up there that's not being used. The second question is, what was the total amount of information found by human review? And that's the area of those colored human review curves. That's less than 20% of the total area of what the computer found. So if information is one of your main goals, it's not being completely captured by current review processes. So you can start asking what happened. The computer, shown in blue again, always gets an answer. If we look at cases where they looked at 1 mixture item, 2, 3, 4, 5, and so on. When they only looked at 1 item, about 75% of the time they'd get an answer. And then as it goes out, they get less and less information. And so let's take a look at why this is happening. What New York did is they classified the cases with a degree of difficulty into simple, medium, and complex. Simple being a 2-person mixture with a known victim, medium, 2 unknowns without any reference, and complex, things that went to 3 or more, partial profiles, or so on. So here are the productivity results. What's the extra effort that people are going through on data that would have been solved if they were using a calculator instead of not using a genetic calculator? Let's look at the simple cases first in green. So there were 35 of these. The computer found about 10 to the 16th on average is the log likelihood ratio information. It had an answer for all of them. And the human yield on these was about 1 in 2, about 50%, which means that the expected extra effort that you can compute is about 2 to 1. So it turns out, how many items do you have to analyze until you get an answer? If there's a 50% yield, then you're doing twice as much work as you have to. When you move in purple in the middle column to the 20 medium cases, then the computer is about 10 trillion, 10 to the 13th, getting answers for everything, and people are finding an answer for 1 in 4 on these 2-person mixtures without a reference, done by RMNE, and they have to do 4 times the work that would be needed based on the data that they already have. And when you go to the 33 complex cases, items, in the 3rd column, the computer is now down to only 1 trillion to 1, 10 to the 12th. The human yield, though, drops to 1 in 5, meaning you expect to do about 5 items before you get an answer, spending 5 times the amount of time, the money on reagents, the delay, all the usual costs that go with processing samples. If you look at an average on the 88 total, the computer averaged about 100 trillion to 1, 10 to the 14th. You see over here, the yield was just under 30%, and on average it's about 3.5 times the effort that was needed. That's sort of the take-home number from the talk, is that on this very diverse data set on mixtures, people are taking 3.5 times the amount of effort to get some score, some match statistic on an item than they would need to if they were just simply using a quantitative genetic calculator. Notice, of course, that as the spectrum of cases shifts to more interesting things, you're going to be closer to 5 times the effort than you are to 3.5. So who benefits from all this? Well, probability is always done in service to utility, starting with gambling 300 years ago and moving to economics and such today. From the laboratory's perspective, it's nice to reduce costs, increase efficiency, and so on. From society's perspective, it's good to get the maximal expected utility from forensic DNA to reduce crime and increase public safety. Different objective functions, but information is good for both. So in conclusion, there's a very growing increase in DNA demand from society, which taxes the lab with workload, complexity of DNA, and so on, with a far smaller increase in the DNA capacity, as you know. The resources are not growing commensurate with that demand, nor the number of analysts. And so automated computer interpretation of DNA evidence is one way forward. And the metrics that we used that I discussed today were objectivity, which computers have if you program them right, ease of use, information extraction from the available evidence, and the productivity that's directly related to the extracted information. I'd like to thank Jamie Belrose, who did the review of all of this data and collection of it, some of our group at Cybergenetics, who was involved in the project, more staff at New York State Police, who gathered quite a bit of the data, and we have a submitted manuscript that we're waiting to hear back from Journal of Forensic Science. And if in the interim, you can download that if you're interested, and there's the URL. It goes into much of what I discussed today, and probably the most interesting thing, it goes through one detailed example showing locus by locus how RMNE loses information relative to quantitative DNA interpretation. Thank you very much.